Okay, well, it's 5 past the hour, so we'll make a start. Welcome to this month's JMCC-ISHR webinar featuring Dr. Julia Liu from the University of Minnesota. My name's Kate Weeks and I'm a social media editor for the JMCC, and I'm joined by Dr. Adam Truen from the University of Melbourne, who will be chairing this webinar. So the way that this is going to work is that Julia will talk for maybe 30 or 40 minutes about her article, which was selected as the editor's choice in the JMCC for August, and then we'll have some Q&A. So if you have a question for Julia, please pop it into the Q&A box, which you'll be able to access at the bottom of your screen. And yeah, I will pop the YouTube link in the chat. So if you want to catch up on any of our recent JMCC webinars, you can take a look at that. And I'll also pop a link to Julia's paper in the chat as well. Okay, without further ado, I will hand over to Adam. Thanks, Adam.
Thanks, Kate, and welcome everybody. Thanks for joining us for today's ISHR JMCC webinar. And it's my pleasure to introduce today's presenter, Dr. Julia Liu. So Julia is an assistant professor in the Department of Integrative Biology and Physiology at the University of Minnesota. Julia completed her PhD at Harvard University, majoring in biophysics, and this was followed by a postdoctoral fellowship at the NIH, during which she was awarded an FI2 Postdoctoral Research Associate Training Fellowship, as well as a K22 Career Transition Award. Julia then joined the University of Minnesota faculty in 2020 and started her lab. And the lab's broadly interested in mitochondrial pathways involved in physiological and pathological stress responses, particularly in the heart. And her lab is currently focused on identifying how dysregulation of the mitochondrial calcium handling leads to adaptive responses over time. So it's my pleasure to introduce Julia today. She'll be talking about her recent work on mitochondrial calcium handling in the heart, and specifically because of her recent publication in JMCC on deletion of the essential MCU regulator, which was selected as the editor's choice. So we'll have, as Kate mentioned, time for questions at the end. So pop those into the Q&A box as you think of them throughout the talk. And without further ado, I'll hand over now to Julia to tell us all about MCU and EMRE in the heart. So thank you, Julia.
Thank you, guys. So happy to be here today. I want to thank JMCC for this opportunity, and also, this honor for Editor's Choice article. And I wanted to also thank Kate and Adam for being up in the middle of the night in Australia to moderate this session. So today, we're going to talk about my paper, actually, and I will say it's actually Hector's paper, and I'll introduce him when the time is right. But we're interested, of course, in mitochondrial calcium in the lab, and I wanted to, as many of you probably know already, mitochondrial calcium is super important in the heart. So obviously, calcium is incredibly critical to the function of cardiac myocytes. I won't be talking much about the cytosol or the ER today, but in this notion of parallel activation, when calcium triggers contraction and the use of ATP, it also goes to the mitochondria to trigger the making of ATP. So it goes in the mitochondria, can stimulate enzymes in the TCA cycle, increases ATP production. This is very good, right, for physiological ramping up the energy output to match increased demand or workload. But it has, a dark side, and above a certain threshold, too much calcium influx is going to open the PTP, and then triggers the permeability transition pore, and which can lead to mitochondrial swelling, which I tried to note here, and trigger cell death, for instance, in ischemic reperfusion or IR injury. And so that means calcium has to be tightly regulated. And one way the mitochondria do that, they control they control how much calcium gets in via this multi-protein complex in the inner membrane called the mitochondrial calcium uniporter. It's it's shown in this cartoon with its many subunits. So MCU is this the pore forming subunit here in blue. And then we have some other sort of accessory proteins in the MICU family, and as well as this protein here, EMRE. And it's been observed that in different tissues, the stoichiometry of the subunits is different, and that can impart different calcium uptake kinetics. So, what does this all mean physiologically? When this was discovered, 50, 60 years ago, this was in isolated mitochondria, and it was sort of an unknown, unanswered question, like, would this really actually matter to a heart or to an animal? And what we had no way to test whether these things that we predicted would be true or not. So you might think that if you inhibit uniporter activity, you might respond less well to physiological stimulation or energy demand. But if you don't have MCU, if you don't have calcium uptake, you can't then open PTP injury opening, and then you might have protection from IR injury. And up until 2011, this was undoable. But in 2011, the gene encoding MCU was identified by two groups, and then the other subunits were also similarly identified. And this really opened up the opportunity to use genetic knockout models to see if these predictions are true. And so I'm going to give a little background on what the initial MCU knockouts were, and this is sort of where things got kind of interesting. So the first MCU global knockout mouse was made with a gene trap method in the Finkel and Murphy labs at NIH, just prior to when I arrived there to work with them. And actually, this is their second paper. I'm a co-author, and Kira Holmstrom and the rest of us used Millar catheters to look at these global knockout MCU mice to see whether the heart contractility really will respond to isoproterenol in a knockout the way it does in a wild type. And of course, you can see that the rise in pressure DPT max increases with iso in the wild types, but the knockouts are not substantially different from the wild type. Of course, they're for statistical reasons, these two bars are actually not significant either. But I would say on the whole, you don't see a substantial dramatic difference in how the wild type and the MCU knockouts act. And I should have mentioned that these MCU knockouts were shown to not have any uptake ability, and they also are protected from swelling. So, and in the first paper, an ex vivo Langendorf ischemic reperfusion assay was also done. And the prediction here, right, if MCU prevents PTP opening, would be that the knockouts should have better RPP, rate pressure product recovery after in the reperfusion period post ischemia, and they should also have a smaller infarct. And this seems not to be true. So cyclosporin A, which inhibits cyclophilin D, a sensitizer of PTP, does protect the wild type. But the MCU knockouts alone don't have that protection. And in fact, the cyclosporin A now also fails to protect the MCU knockouts, indicating something is perhaps different in that cell death pathway. And so this was a little bit of a conundrum. So, why is this MCU knockout mouse not behaving the way we thought MCU knockout should? And to maybe clarify, or, in 2015, the two groups, the Elrod and the Milkington labs, using the same MCU floxed mouse model, published back-to-back papers, in which they took the MCU floxed, newly developed MCU floxed mice, crossed them to the alpha MHC mercury MER to make them cardiac specific as well as tamoxifen inducible. And so now in adult mice, if you use tamoxifen to knock out MCU, again, no more calcium uniporter activity, which I haven't shown, but in their catheterization experiments with isoproterenol, this is from the Luongo paper, the two control groups, of course, see an increase in DPDT max, and which is much blunted in the MCU knockouts. And correspondingly, then when they did a ischemic perfusion injury experiment, in this case, in vivo, so with LAD coronary artery ligation, they noticed that, of course, the area at risk versus LV area is not changed in their experimental conditions, but the infarct area relative to the area at risk does decrease in the knockouts, showing a protection from IR injury. So now we have, you know, the expected or predicted results with MCU knockout. So it's at least true that if a mouse has a functioning MCU and then at some point has that deleted, they do show what we would expect. So just to sum up these chronic versus acute MCU deletion results, in the global MCU knockout, where MCU is gone everywhere, all the cells forever, we don't see substantial changes in the knockout's ability to respond to adrenergic stimulation. We don't see any protection from IR injury. With the Floxed model in the later papers, which is cardiac specific and inducible, we then see a blunted response to adrenergic stimulation and a smaller infarct. And I should also mention that there was the Mark Anderson's group also made a mouse that was heart specific, but also in the germline using the alpha MHC, no CRE, CRE, but not tamoxifen inducible. And they do see this result, but they also see this result with no, sorry, my pointer's not working. Oops. Is my pointer working? Anyway, they don't see the protection past, thank you. So it is? Okay. Maybe I should put the pointer options. Okay. So so that also, I should mention two things. One, the MCU knockout mice are in an outbred CD1 background, because when the global MCU not deletion appears to be embryonic lethal in Black 6 mice. Even in this outbred background, fewer mice are born that are knockouts based on predicted Mendelian ratios. So fewer than a quarter of the mice from heterozygous crosses. Also, the injury, the IR experiments here, including in the Anderson group where we don't see it, where there's no difference in infarct size, were done in ex vivo Langendorf. These experiments by these two groups were done in vivo. So there's a slight difference there. Okay, so I'm going to go back to here now to introduce where I really enter this story. As I mentioned, their 2014 is when I started my postdoc, and we were interested in knocking out different members of the calcium uniporter to see what they really do physiologically. And I made knockouts of EMRE as well as MICU1. I'm going to concentrate on EMRE today. And I want to introduce EMRE, this protein, in full. I think it's a very interesting protein. So it was discovered by Vamsi Mutha's lab in 2013. And cell culture was shown to be essential for MCU and mediated calcium uptake, hence essential MC regulator. And recently, a cryo-EM structure of the human MCU was published together with EMRE. So the different colored proteins here are MCU. It forms a tetramer. And here's the transmembrane domain, and that's where EMRE binds. EMRE is shown here in pink and 1-to-1 with MCU. And so the idea is that when bound to EMRE, MCU adopts an open conformation that allows it to transport calcium. And as I said, the stoichiometry and even the composition of the uniporter varies in different tissues and even across organisms. So if we look at a phylogenetic tree, we can see that many, but not all, eukaryotes possess an MCU homolog. So these everything with a dark red here has MCU. And some of those, but not all, have an MICU1. So one of the regulators, the other intramembrane regulators here in green. And EMRE is probably the most exclusive. It's sort of only here in the complex, so-called complex eukaryotes, the metazoans, which includes the vertebrates. And I'm oversimplifying the evolutionary history of EMRE here, but the question in my mind, at least, especially back in 2014, was, what would these so-called higher organisms stand to gain by having EMRE? Does EMRE regulate, the uniporter in some special way that you don't have with just MCU? Why is it there? And so we went and made a germline EMRE knockout mouse. And at the time, right, CRISPR had made making these transgenic mice much faster. At the time, also the cardiac specific MCU knockouts hadn't been published. So we went ahead and we made the global CRISPR mediated EMRE knockout mouse. You can see in isolated mitochondria, we don't get calcium uptake. So some of you, if you're not familiar with reading these calcium green diagrams, basically what we're doing is we're measuring calcium in the buffer outside the mitochondria. So each time you add calcium and dignified arrows, normal mitochondria will uptake that calcium, taking it away from the dye, and you'll see this exponential decay. Do this enough times and PTP opens, all the calcium is released. The EMRE knockouts and the MCU knockouts previously show stepwise, which means that each time you add calcium, it just stays in the buffer because the mitochondria can't take it up. Okay, and similarly to the MCU knockout mice, EMRE knockouts also were only viable when outbred, and they had a much decreased birth rate. So they, if you expected, a quarter of these knockouts in a heterozygous cross, you actually get only about a tenth or so. So this is, and then what happens? Oh, sorry, forward. What happens in the heart? Those two things we mentioned: response to stimulus, protection from IR injury. We saw that when in this experiment we were measuring ejection fraction by echo and injecting isopaternal in IP, intraperitoneally, and we see that the wild type and the EMRE both respond quite well to ISO. And we also saw that in an ex vivo Langendorf IR experiment that the infarct area of the EMRE knockouts versus the wild types is pretty much the same. And just like the MCU knockouts, the wild type are protected when cyclosporin A is present, and then knockouts are not. So we can add, this is sort of what's not jiving here, right? We don't know why this is happening. And our guess is maybe it's sort of compensation that's happening for uniporter when the uniporter is absent, in those surviving mice. And so I'm adding EMRE now to our, schematic. And we're going to return now to the question is, what is this compensation? Is there compensation? When would it occur? So does it have to happen, let's say during development, right? During the embryogenesis period where we see embryonic lethality. And if you get past that point, the surviving mice are then rewired. So, and also is this, would this, along the same lines, would that compensation be just a matter of waiting long enough, so time is sufficient, or again, is it developmental? So we thought we could answer sort of two questions at the same time. One would be, what would happen if we make this EMRE deletion in the cardiac specific cardiomyocytes in adulthood with tamoxifen-induced interval system? And also, what's the time scale of this compensation for losing uniporter function? So what we did then is we went ahead and we crossed EMRE that floxed mice, which we obtained from a consortium, the MMRC, to these heart-specific, the alpha MHC mercremer tamoxifen-inducible cream mice to make conditional EMRE knockout mice. And we will then, in young adults, inject tamoxifen to delete EMRE, and then we'll wait either a short-term or a long-term time scale of deletion to see what happens. And so here's a picture of Hector, who took on this project. And so we'll first go over what happens in this short-term or acute period, 3 weeks after tamoxifen. So as we, you might expect, and what we obviously had to verify, EMRE protein levels do decrease right? In the knockouts, so our two control groups are the mercury-mer only and the flox groups only. And we have less EMRE. Interesting, we don't have any changes in MICU1, but we do see less MCU. And so here's the protein expression of MCU, which is also decreased substantially. And we also ran blue native gels to look at the complex without denaturing it. And you can see MCU is sort of a smear. Often you will see it in either the 480 and often in a higher band corresponding to the MCU uniporter complex. And in the EMRE knockout, this is the blue native run and then probed with MCU antibody. So MCU actually shows up in definitely something much, much smaller, wherever that is. Interestingly, that decreased MCU it's at the protein level, not at the mRNA level. So of course, mRNA is gone because we just deleted the gene. MCU mRNA is stable. So that decrease in MCU is likely mediated through protein stability, we think. Okay, and I should also mention, I've concentrated on calcium regulation in mitochondria through the uniporter. There are also efflux pathways for calcium to leave the mitochondria, LATM1 and NCLX being the best known, and these are not changing at the protein level after EMRE deletion. The mice also look very normal. Their body weight doesn't change, their heart rate doesn't change. So altogether, you wouldn't know by looking at them that they're EMRE knockouts. But if you take out the mitochondria, we took out isolated cardiac mitochondria and then did that calcium retention capacity assay, and you can appreciate that both the control groups can take up many boluses of calcium before PTP happens, and the EMRE knockouts don't take up calcium appreciably. And along those same lines, if you measure the absorbance of mitochondria and then add a large amount of calcium, enough to open PTP, when swelling occurs, absorbance drops in the control groups to much less of an extent in the EMRE knockouts. And we also sort of followed this up with a membrane potential assay using safranin, which is a membrane potential dye. It actually, so when the increase of fluorescence of this dye corresponds to the depolarization of mitochondria. So when we add that high dose of calcium, you can see it's stable in so it's stable in the knockouts unless you also give it FCCP to depolarize, whereas the knockouts rapidly, like the swelling assay, will undergo mitochondrial membrane potential depolarization. So, okay, that's showing that when we knock out EMRE, we've basically eliminated uniporter function. It doesn't uptake calcium, and they also seem to then be protected from PTP opening. And we also looked at sort of basal intramitochondrial calcium content. It's not, these are not the easiest assays. I think everyone has their least disfavorite way of doing this. We took out mitochondria, loaded them with fluo4, and then calibrated to high, maximal and calcium and low calcium. In this assay, what we were able to show is relative to the two control groups, the EMRE knockouts seem to have less basal mitochondrial calcium. And to corroborate this, we also looked at the phosphorylation of PDH. So PDH is one of the enzymes that calcium in the mitochondria regulates. So what it actually does is it stimulates PDH phosphatase to remove that phospho group, and the non-phosphorylated form of PDH is the active one. So actually, high levels of phospho-PDH indicate that there's actually lower calcium in the mitochondria. And so what we have here is, again, in the knockouts, an increase in phospho-PDH, which is consistent with a decrease in mitochondrial calcium. Okay, so we went ahead then to figure out if this short-term EMRE deletion affects the matching of energy supply to demand, right? That's the question here. And in mitochondria, if we measure ATP production rate using a luciferase-based assay, providing ADP, we've noticed that in the presence of calcium, then the rate of ATP production increases in both the controls. And this is much attenuated when in the EMRE knockout. So the presence of calcium doesn't appreciably change the ATP production rate. And we then did that kind of echo experiment in these mice with short-term EMRE deletion. And after injecting isoproternol and measuring over time, these experiments were done by Michael Zhang and Tim O'Connell's lab, who was blinded to the mouse genotype. And we see that the ejection fraction responds right to ISO in the two controls, and that response is blunted in EMRE knockouts. And I should point out that these are standard deviations, not SEM. Just that's my particular shtick with statistics. So up to now, our data are agreeing, right, with the idea that mitochondrial calcium uptake is important for matching ATP supply to demand. What about IR? We then performed that Langendorf IR. This is in collaboration with Dwayne Townsend's lab, experiments performed by Jackie Stevens. And we did, this is the short-term EMRE knockout. And across these different measures of function, so rate pressure product, dPDT max and min, after reperfusion, relative to the right after ischemia, you can see that the function of these hearts, the knockout hearts, is let's say less dysfunctional than it's protected relative to the black and the purple, the two controls. And so they have better preserved function. And if you look at creatine kinase release as a marker of the damage, the EMRE knockouts in green have less damage. So, and we also measured, we have an undergraduate who helped us analyze these TTC stains for infarct size. In ex vivo, of course, the entire heart is the entire LV is the area at risk, and we can see that the EMRE knockouts have less of that white staining, meaning that their infarct area at least shows a trend towards smaller infarcts. So overall, these data support that preventing mitochondrial calcium uptake is protective during IR. And this jives well, right, with the MCU knockouts in the Allred and Wilkinson lab. So whichever way you do it, when you get rid of mitochondrial calcium uniporter function acutely in an adult mouse, they have the results that we'd expect: blunted response to isoproternol or adrenergic stimulation, and also protection from IR. Okay, so now to get at that question of, well, is there an adaptation that slowly gets rid of these things? And does it just, if you wait long enough, is this all a function of time? We used then the same mouse, the same breeding and injection strategy, and waited 3 months instead of 3 weeks, and we'll call this the long term. And in the long term, we again checked that EMRE and MCU levels are still down. I seem to have omitted the blue native gel here, but again, the results look very similar to the 3-week. At the mitochondrial level, when we take them out and perform calcium retention capacity, you can see that, again, not very good, not much at all calcium uptake when the controls do quite well. And in swelling assays, the controls swell and the knockouts to much less of an extent. And if you we checked again to see whether we may have, well, maybe mitochondrial calcium can slowly recover back up to basal levels. And in our hands, this was not true. So if we do our flow 4 assay again on mitochondria, we can see that the intramitochondrial calcium levels seem to still be, down in the EMRE knockouts. And if we look at phospho-PDH, it's still up, actually, in the knockouts relative to the controls. So we think that intramitochondrial calcium levels are still reduced even with long-term absence of EMRE. And so the question, right, then what about ATP production? And in that same experimental condition where we in the absence and presence of calcium, the two control groups do show a response to that calcium in an increase in ATP production rate, whereas the EMRE knockouts in the long term still don't seem to really increase ATP production when stimulated by calcium. Okay, so then the big question next is what happens in the heart in terms of the response to adrenergic stimulation via echo versus IR. And so in the echo experiment, what we saw was what resembled the 3-week data quite maybe slightly less of a bluntness, but still a bluntness of the knockouts relative to the response of the controls to isoproternol. So that was saying, okay, just waiting long enough doesn't get you, doesn't buy you, you know, a renewed response to isoproternol. But sort of counterintuitively, our ischemic reperfusion experiment showed that now the green line is falling right on top of the blue, black and purple lines. That means that these hearts no longer have any better function in that post-ischemic reperfusion period. There's no significant difference now in creatine kinase release. So the protection against IR that we saw short-term after EMRE deletion seems to disappear as a function of time. And so then, you know, we had these interesting results. We said, but why? Credit due to JMCC and the reviewers who actually really did also push us to ask more of the but why, and had some very good suggestions for what to look at. And so we're going to look through some of the potential candidates. And, you know, maybe there is a known link right between MCU and CAMK2 in the cytosol. If we look at whole heart lysates here, however, we don't see any difference in the phospho-CAMK2 levels relative to total CAMK2 in our EMRE knockouts. And these are from, I believe these are from the 3-week, but I will say that just to not overload slides with a lot of very similar looking data, this is also true in the long term. So we don't see a change here. What about cyclophilin D, right? So this is our sensitizer to MPTP. So it's because the MPTP pore is still a controversial subject, but everyone agrees that cyclophilin D does sort of promote MPTP opening. Maybe that the levels of the cyclophilin D are changing, and we don't see a change though in our EMRE knockouts at either the short or long term. Okay, what about maybe when you have no mitochondrial calcium uptake for a long time, you've somehow changed how your respiratory chain or how well you can consume oxygen to make ATP. So we did some seahorse assays using 2 different substrates, so pyruvate malate and also palmitoyl carnitine and malate for fatty acid oxidation. And we looked at the basal oxygen consumption rate as well as state 3, state 4. And after the addition of ADP and also then after ligamycin and also after FCCP. And we saw that the respiratory control, we also measured calculated from this the respiratory control ratios, and we did not see differences in the EMRE knockouts at either the 3-week or the 3-month time points. And it did not seem to matter which substrates we fed the mitochondria. So to corroborate that we don't see evidence, at least in these assays, of a metabolic switch, we measured the levels of CPT1B, right, the rate limiting enzyme to transport fatty acids into the mitochondria, and we don't see differences there either at either time point. Okay, but there, another reviewer suggestion was to look at complex 1 activity, and I encourage you, there's a great paper from the Chaudhary lab at showing a link between complex 1 and MCU, and in fact, that they interact in the inner membrane. And so we measured using these are sort of plate reader assays in frozen mitochondria. So what we're looking at is really sort of the maximal possible complex 1 activity, or complex 1 or complex 2 activity when we feed it all the substrates it needs and all of that. So in the short term, we didn't see a difference between our control and our EMRE knockout. In the long term, we see this trend, and it could be that with prolonged absence of uniporter function, that complex 1 is no longer able to be as active. However, we didn't get statistical significance here, and we did not have more mice to do these long-term experiments on. So there's probably more to be uncovered here, and I'm very excited to keep doing that. Okay, and then the last sort of potential why candidate was, what about mitochondrial ROS, reactivate oxygen species, right? So we know that calcium is a trigger for the opening of MTTP. It's thought that ROS can synergize with calcium to sort of potentiate the opening of MPTP. We should look at ROS production. We looked at ROS production actually with this Amplex red assay, again in isolated mitochondria. So this will detect H2O2 peroxide. And just as an example of how the assay looks, so if you put an EMRE control versus knockout here, and then we here we added ADP, and then we can actually show also that this assay works because controls like antimyosin A, which is a positive control, makes a lot of ROS, versus DNP, which slightly depolarizes and actually decreases ROS production. So this is how the assay works. Our results, when quantified, show in the short term or the long term, there wasn't a substantial or observable increase or decrease in ROS production in our EMRE knockouts relative to control. However, we also looked at protein carbonylation, which is a marker of damage from reactive oxygen species on proteins on mitochondria. And so the darkness of all these bands sort of correspond to the carbonylation levels. And you can actually see that in the EMRE knockouts in the short term, that the carbonylation levels seem to be decreased. Again, compared to Phlox, this is not significant. However, it is there is a significant difference between how much carbonylation there is in EMRE long-term knockouts versus EMRE short-term knockouts. So our sort of leading candidate that we're hoping to pursue further is what is happening to the balance of reactive oxygen species, the production, and also the scavenging of the antioxidant capacity of mitochondria, provided that you leave them for long enough without the uniporter activity. So to sum up, this is our Black 6. I should have mentioned that EMRE Phlox. This is a model with cardiac specific deletion of EMRE. We seem to block calcium uptake into the mitochondria. And in both the short-term and long-term, actually, these mitochondria are not capable of taking up calcium through MCU. Their levels of intramitochondrial calcium are also reduced. But without this functioning uniporter right at the 3-week time point, we see that they have a attenuated ability to respond to isoproterenol in terms of their increase in ejection fraction. In 3 months, this appears to still be maintained, which might mean that the compensation that we saw right in the global MCU and EMRE knockout mouse, where they had virtually no problem acting like wild type and increasing ejection fraction could be something that happens in these mice that don't have MCU during embryogenesis, potentially. This is a speculation. In terms of IR injury, the rate pressure product we showed in this ex vivo model shows that the knockouts do have better maintenance of cardiac function post IR, so protection. But now waiting that just a few more weeks later, that response is transient, and we go back to being not protected from IR injury. And so we're very much interested in sort of if the cell death pathways triggered in this IR response are different. Are they possibly different also because the state of reaction of oxygen species and oxidative damage in the mitochondria are also changed? Okay, so with that, one more thing I forgot to mention earlier. So we also have discovered, like here, the difference between the Elrod-Wilkinson models and the Anderson-Finkel-Murphy models of global MCU knockout some were done in vivo and some were done ex vivo, because in this example, in our EMRE short-term knockouts, they were protected even in an ex vivo experiment. I think we can rule out that the modality of inducing IR is not what's causing the difference. It's something about the actual biology of the mouse here. Okay, and so I think it's, the burning questions that my lab are so interested in, of course, is what are these time-dependent adaptations to mitochondrial calcium uniporter dysregulation? We also want to know whether maybe these adaptations, of course, it seems to be a little bit maladaptive here. Is that because maybe there's other beneficial, is there a benefit to this adaptation? How does this maybe manifest in different types of stress conditions? So that's where our lab is going next. And with that, I want to thank everybody who was involved in this work. So everyone in blue is an author on this JMCC paper. And of course, Hector led this work. And I realize now I really should have actually put Hector and JP under formerly lab, because they both have recently moved on. JP has graduated with his PhD, but maybe I don't like to see you go. You guys have very big shoes to fill. We're hiring, and in case anyone's interested. And lastly, thank you so much to JMCC for handling this paper. We got excellent, very, very thorough and thoughtful and extensive reviews back from our reviewers, but I think they did make the paper better, and I'm just honored that it was selected as the editor's choice. And so thank you all for listening. I'd be happy to take any questions.
Thank you very much, Julia. That was fantastic. Really enjoyed that. So feel free, everyone, to put comments in the Q&A box. And while we're waiting for some of those to come in, maybe I can get started with a couple that I had. So I was wondering, so you mentioned that with the EMRE deletion, you saw obviously the decrease in the protein level, then the mRNA level was down as well. But with MCU, there was the loss of the protein, or a decrease in the protein, but a maintenance of the mRNA level. So that was really interesting. I was sort of thinking about the MCU loss of protein, and you mentioned the protein stability. But then what about with the MICU1? Because I guess that's structurally related to on all part of that complex as well. And that wasn't decreased in protein level. So yeah, I'm just wondering if you can comment on why that might be, or
yeah, I think it's that's a really good question, because of course, our Blue Native says that the complex is not the normal MCU complex anymore. So if there's still MICU1 protein, what is it doing? So we did not probe our blue native gels for MICU1, unfortunately. That antibody is a little bit trickier. But I will say that there are papers that say that MICU1 is involved in other non-uniporter functions. So in the maintenance of cristae and in the mycose complex. So there's potentially other things that MICU1 is doing. And so I do think it is quite a sort of unanswered question that I hope so maybe the structural biologists and the biochemists will really get at and hopefully share with the rest of us so we can also figure it out. Yeah.
Can I ask a question?
Yeah, of course.
That was fantastic, Julia. I really, really enjoyed that. And I love your enthusiasm for EMRE. That really came through. So I know the focus of this paper was very much on what's happening in the mitochondria and how energy demand is meeting supply or not meeting supply. I'm just wondering if you've looked at all on the effects of EMRE knockdown on calcium handling in the cytosol. And I guess I was really interested by the functional data you showed with the response to isoproterenol. Are you suggesting that that is because the mitochondria aren't producing enough ATP, or are there effects on cytosolic calcium levels that are somehow affecting excitation, contraction, coupling, and the response to ISO?
That is such a great question. So I think maybe a year ago or so, I would have just been like, well, I think, it's been shown that the concentrations of calcium that get taken into mitochondria are negligible compared to what's in the cytosol. It should not really affect cytosolic calcium at all. But I'm beginning to question that myself. Actually, we in our MICU1 knockout, we do see that potentially cytosolic calcium is affected. So I can no longer say that I don't think this would matter. I don't know that it actually affects it on directly though. I don't know that I would say, well, it's because the calcium can't go into the mitochondria anymore, so it actually changes what's happening in the cytosolic pool. I don't think that's directly. I think what would happen is the mitochondria are sensing we have messed up calcium regulation. We need maybe we're not making ATP at the right times like we should be or whatever, and this then goes and changes. We see the phosphorylation of PDH change. I think it could be that there are these retrograde signaling pathways that say the mitochondria are stressed, and they're telling proteins in the cytosol or even mRNA that we need to sort of adapt in some ways, and that I think is still very, very much unknown. To answer your question directly about the ejection fraction and in the echo, I do think our assumption is that we're asking the heart to work harder, but the mitochondria aren't getting the signal to make more ATP. So, they're not. But that's as much as I can say. I don't. I think it is a very, very, we haven't looked carefully, we're just actually getting the ability to do ion optics experiments on adult cardiac myocytes. So something we hopefully will look into in the future.
I think that'd be really interesting. The chem kinase phosphorylation wasn't changed, so I guess that sort of tells us something about what calcium may or may not be doing in the cytosol. But yeah, I wondered if you'd looked at CIRCA or the NCX or something like that, and if their activity was altered to try and counteract the loss.
I would love so going protein by protein and also just the overall levels, it seems to me I don't know if I would call it brute force, but just there are limitations, right, to doing it. The information you get from a Western blot. So I don't know if, I think one possibly we might go for is to just sort of do an overall like unbiased approach like proteomics, just to like especially phosphoproteomics, just sort of see what we see, because otherwise we're the ones selecting which proteins to look at. As people, like, you know, there's the things we know, the things we don't know, and the things we don't know. Like I maybe we're not looking at the right ones yet. But also, I mean, in terms of really getting a more finessed or you know, detailed grasp on, like, not just a snapshot of protein levels, we may have to you know, go for, hopefully there's a lot to do sort of maybe sensor wise in living cardiac myocytes. But maybe the proteomics will tell us like what the next targets should be to look at. So yeah, things we're thinking about, but really great question, Kate. Hey,
I've got another one. Just thinking about the, I guess, endogenously, how is EMRE transcriptionally regulated? Like, what transcription factors are involved, or, you know, and is it similar to MCU, which I think has been shown to be under control of
that is known. It's something that I should be asking you. So maybe you'd like to, we can talk more. But yeah, so interestingly, and let me think. This is going back to data that I from my postdoc in the MICU1. When we made MICU1 global knockout, we did see part of our huge, like, what helps these mice adapt to calcium overload, which is what the MICU1 deletion gets you, is that EMRE starts to go down. And there, I forget in which tissues, but maybe liver, I think we saw that EMRE mRNA was going down. Later, Jay Hui in our lab, and hopefully we'll get this paper submitted soon. When we make cardiac specific knockout of MICU1, this is in using Yuri Hejnotsky's MICU1 flox mouse. We don't, in the heart, in the cardiac myocytes at least, we don't see a decrease in MICU1, EMRE mRNA. So there at least in the heart, in this context, we think a lot of EMRE's protein expression is regulated post-translationally. So there's degradation pathways through these MAAA proteases. So because we've been concentrating that, we haven't gone back to look at what might be regulating EMRE transcription, since we don't see changes in the heart. But I think it's still, under some conditions, I think EMRE transcription can be affected, and I don't know what regulates that. It's a good question.
Yeah. Another one I had was so I think with just trying to think back to the data you showed on the slides, with the longer term experiments, you did some seahorse experiments, I think, showing that there was no difference in the oxygen consumption with the knockout. Is that right?
Yes.
Yeah. So, and that was, I just wanted to clarify, was that on isolated mitochondria?
Oh yes, I should have said that. Yeah, that is isolated mitochondria.
Okay. Yeah. Yeah. So, well, okay. So I guess the follow-up question for that is, had you looked at any markers of mitochondrial density in the tissue, and does that change over time? Mitochondrial content.
Yeah, good question. I think we haven't noticed when isolated mitochondria that there's a difference, but it is a very good point that maybe we did not do like an EM, or, you know, even look at mitochondrial density in cardiac myocytes. So that is something to verify potentially, right? But in this, of course, we normalize to, like, you know, micrograms of protein, mitochondrial isolated protein. Yeah.
All right.
Well, I just jump in with Oh,
Yeah.
sorry. Can I just ask
Go on.
one final question?
Yeah,
If that's okay. So you talked a number of times or alluded to some compensatory mechanisms that might be happening. Have you got any clues or ideas what those might be?
Yeah.
Or where are you going to start to try and
Where
figure
are we
that
going to
out?
start? Yeah, that's a really good question. So I think what for at least in terms of this paper, the EMRE, I think our clues that seem at least the most promising at the moment, right, are to look more carefully at what's happening with complex 1, look more carefully at what's happening with ROS. So what I thought might fit, which I'm not sure it does, but our initial thought was, okay, maybe if you can't get calcium into the mitochondria, then you don't stimulate ATP production as much. So maybe your, respiratory change gets kind of relaxed, doesn't make as much ROS. And so then you start over time, like being like, well, I don't need my antioxidant capacity anymore. And you start decreasing those proteins. But then I would have thought, if that were true, I would have expected like, okay, ROS carbonylation would be the same at the short term, but then get worse in the knockouts at long term. And that didn't seem to be exactly what we were seeing. So we're still trying to, so we're probably going to have to, when I wasn't originally a ROS expert, I might have to start digging a little bit deeper into that literature and talking to potential collaborators to try to get that sorted out. And if it is true that complex 1 and MCU's interaction is being, modulated, it could mean, right, that the rate of ROS production from complex 1 is also changing. And so, but right now, if anyone would like to take Hector and Jeffrey's place, please come to our lab and we'll figure out how to do these, make like useful measurements, and we'll get that sort of pinned down. But yeah, those are our best guesses for now, but they're still speculative. Yeah. Okay.
Well, I think we might be just about out of time, so we'll leave it there. So thank you very much again, Julia. That was a fantastic presentation. Really enjoyed it. And yeah, thanks again. And we'll enjoy the rest of your day. And
yes,
we'll
you
see
too.
later.
And please go back to sleep.
Yeah.
I hope you can. Thank you guys for so much.
Thank you, Julia.
Thank
That
you
was
very
really
much.
great presentation.
Okay.
Thanks,
Yeah.
everyone.
See you.
Bye-bye.
Bye.